Welcome to Dr. Baliga's podcast for the Curious Doc. Today is December 17, 2025. This video is on a Cell paper published last week, December 11, 2025, titled Protein Restriction Reprograms the Multi-Organ Proteomic Landscape of Mouse Aging. Protein restriction is a dietary intervention shown to modulate aging-related nutrient sensing pathways, including insulin-like growth factor, IGF-1. It's a mechanistic target for rapamycin, and AMPK, that is adenosine monophosphate activated protein kinase axis. Study design: multi-organ proteomic atlas of aging in mice combined with dietary protein intervention. 41 organs were collected across 8 lifespan time points, 1 to 24 months. 2,565 tissue samples with 3 to 4 biological replicates per organ were obtained. Intervention: 3 months of dietary protein restriction at young mice, 7 to 10 months, midlife 12 to 15 months, and old age mouse 20 to 23 months. Diet composition: low protein 5%, moderate protein there was 20%, 20.7%, and high protein 40% of total energy. Fat was fixed at 16.5%. The human cohort: 272 adults aged between 26 to 88 years with paired plasma samples collected in 2015 and 2018, a total of 544 samples. The total plasma samples, 544, were analyzed by proteomics assays. Exposure: change in percentage of dietary energy derived from protein. Clinical variables assessed include insulin, hemoglobin A1C, LDL cholesterol, and blood pressure. Statistics: analysis of variance with Benjamini Hochberg false discovery rate controlled below 0.05. Human analysis used linear mixed models adjusted for total caloric and lipid intake. What did they find? In the mouse study, aging altered more than 11,000 quantified proteins with strong organ specificity. Protein restriction reversed age-associated proteomic trajectories in adipose tissue, pancreas, brain, kidney, and cardiovascular relevant pathways. Midlife intervention showed the strongest systemic benefit across organs. Brown adipose tissue activation: uncoupling protein 1 increased approximately threefold low protein diet compared with high protein diet, p-value less than 0.01. Signaling effects: there was reduced IGF-1 signaling and increased AMPK phosphorylation across multiple organs. Human studies: lower protein intake was associated with reduced insulin, reduced hemoglobin A1C, reduced LDL cholesterol, and systolic blood pressure. Inflammation signal: very low protein intake was associated with increased inflammatory proteins, including pentaraxin 3. Limitations of the study: female mice were underrepresented. Survival outcome not directly measured. Extreme protein restriction was associated with hepatic steatosis in mice. The key takeaway: modest protein restriction, particularly in midlife, confers multi-organ metabolic and cardiovascular benefits, while extremes may be harmful, that is, too much protein or too little protein. Clinical pearls: protein quantity influences aging biology independent of total calorie intake. Midlife dietary interventions appear more effective than late life initiation. Benefits are mediated through brown adipose tissue activation, insulin sensitivity, and insulin-like growth factor IGF-1 suppression. Human data support cardiometabolic benefit, but caution against excessive protein restriction. Clinical translation favors moderation rather than elimination. These are some eponyms related to this study. Hit pause and read at your own leisure. If you enjoyed this episode, share it with your colleagues. When we change how we see disease, we change how we shape lives. Until next time, stay curious, stay compassionate, and keep seeking wisdom. Podcast brought to you by masterbedfacts.com. This podcast is for physicians and healthcare providers. If you enjoyed this video, please like, subscribe and forward to your friends.